Hello everyone, welcome to another lecture for drug delivery engineering and principles. We are now talking about micro and nanoparticles. So, just a quick recap of what we did in the last class. In the last class we introduced what are micro and nanoparticles. So, these are essentially the miniaturized versions of your matrix devices. So, fairly small, anything less than 100 micron is microparticle, and if you are talking about nanoparticle we are saying even less than 1 micron. We talked about what are the advantages over macro devices. So, several of them we talked about. First is of course that you do not have to do any surgery now, you can just directly inject it. You can get more targeting, you can get more sort of leverage with them. So, if you want to inject it in a sensitive organs, you can find a space, you do not have to do that surgery, and patient compliance is very high, and then several other things we talked about. And then we discussed how they interact with kidney. So, kidney is again one of the major organ that decides what is the residence time of these particles. And so, first we looked at what size ranges these particles flow through kidney and what it does not. So, first thing we talked about size, and what we found that anything which is greater than 6 nanometer, anything greater than 6 nanometer will not be filtered, just because the GBM, the globular membrane, the basement membrane is not going to let it pass through. Then anything between 1 to 6 nanometer, so anything between 1 to 6 nanometer, these will pass through quite rapidly, because the basement membrane is fairly permeable and the 1 is going to pass faster through the 6, and then there is really no interaction. But once you go below 1 nanometer, what happens is the glycocalyx of the cells in the surrounding will start interacting with this, and eventually this will prevent the faster clearance. They will start interacting and will have a much more tortuous route through this glycocalyx. So, these were some of the things we discussed in respect to kidney. And then we talked about charge. So, this again this GBM is negatively charged, and so that means that if there is a positive charge particle, it has quite a fast attraction towards it, and so the positive charge particles get cleared faster, and the more the ionic the particle, the less is the clearance through the kidney. Okay. So, let us continue and let us define some terms for particle mediated delivery. And so, the first thing of course is particle size and shape, and these are really nothing but defining what is the size and the shape of them. So, how big they are, what sort of morphology they are, whether they are spherical, whether they are rod shaped, whether they are disc shaped, so whatever shape they might be. And of course, there are several techniques that you can use to sort of get an estimate of this. So, you can either get some sort of a qualitative estimate. So, you can use microscopy. We have now very powerful microscopes that can image even 10 nanometer and lower. So, and you can get down to even electron microscopes that can now even go down to single wavelength, single nanometer size ranges, single digit size ranges. And then you can do this quantitatively. So, there are several techniques, there is Coulter counting or light scattering techniques that will sort of scatter light, and you can kind of get an estimate of what is the size that is scattering it. You can get a distribution of what sort of diameters are present in the whole population, maybe that not all particles are similarly sized, and all of these can be used to sort of get some sort of an estimate of the particle size and shape. Another is polydispersity. So, it links to this average distribution, and what it is in unlike these macro devices where you have quite a bit of control as to what is the size of the device you are making. If you are making 1 millimeter, you typically get it within plus minus 0.1 millimeter, but with these micro devices and nano devices, that is not the case, because and we will discuss about why there is polydispersity, but then there is quite a bit of polydispersity that you may find in different samples. And to define that, we have this term called polydispersity that basically calculates the size distribution. So, whether how many particles are there with, let us say 1 micron, how many particles are there with 500 nanometer, and all this will sort of define what polydispersity is, and essentially it is just a measure of how broad or narrow the size distribution is. And then the carrier composition. So, first of all, what is the polymer that you are using, what amount of polymer is that you are using. So, how much percentage is polymer, how much percentage is drug, how much percentage is other components like solvents and surfactants if they are there, if there is any other additives that you are adding. So, all of that is present in that case. So, some more definitions. Next thing is the encapsulation efficiency, and that comes in when we are sort of talking about drug itself. So, what is it is how much of the drug that I started with I was able to encapsulate in my polymer. So, let us say if I want to sort of make particles and I want 1 milligram of drug to be encapsulated in these polymers particles. So, I make these particles and we will discuss about various synthesis methods. So, let us say the particle synthesis is done, and at the end of it I end up with 100 milligram particle. Of course, this 100 milligram is essentially the weight of the polymer plus the drug, and then what I do is I then dissolve this whole 100 milligram of particle and see how much drug was actually there, and I found that instead of 1 milligram I could only get about, let us say, 800 microgram drug in this 100 milligram. So, that means I have lost about 200 milligram of drug during the synthesis process. My encapsulation efficiency is going to be nothing, but it is going to be 0.8 milligram divided by the initial drug, which is 1 milligram, multiplied by 100 to get a percentage, and that is nothing but essentially 80%. So, in this case my encapsulation efficiency is 80%, but this is how it is going to be typically defined. Okay, what about loading level and encapsulation ratio? So, this is another way to define to get an estimate of how much drug is present, and what it is just the weight percentage of the drug in the particle formulation. So, if I just take the last example and I said I had 100 milligram polymer and that was containing about 800 microgram of drug. So, in this case now my loading level is nothing but 0.8 microgram or 0.8 milligram divided by 100 milligram, and so my loading level is actually less than 1% in this case. So, that is how it is sort of defined, right. I mean, so if I multiply this by 100 I am going to get 80 by 100. So, my loading level is only 0.8%. So, that is how it is typically defined. And then we have stability. So, whether how stable the particle is, whether I can store it for longer durations or not. I mean, this may have nothing to do with the drug itself, or it is a combination of drug and the polymer stability, but maybe my polymer itself does not really remain stable. So, the drug will obviously come out. So, essentially chemical stability refers to as the stability of the drugs inside the particle over time. So, what environment you are storing it in, what are the different conditions, and the physical stability is essentially what if the particles are degrading, they are absorbing, let us say, water or moisture from the air, and then essentially causing them to sort of degrade and erode over time even before it is put in the body. So, that becomes important in terms of determining the shelf life and efficacy of these particles. Okay, so briefly I had mentioned in the previous class that these particles can also be used for intracellular delivery, and so what do we mean by that? So, let us say this is a cell that we have a picture of, and so cells itself have sort of evolved different pathways through which they can take up external material. This could be glucose or any sort of energy that they need, or could be food for some pathogenic sort of cells. So, there are several of the ways through which they take large molecules like particles. This could be phagocytosis, mostly present in immune cells, and then rest of the other pathways that are defined here, these are shown by nearly all types of cells. So, this could be macropinocytosis, which is nothing but the membrane ruffling. So, the membrane will ruffle and just sort of eat up whatever is in the surrounding. It could be mediated by some sort of proteins present on the cell membrane. So, this could be a clathrin protein, and so they form small pits, or this could be caveole proteins, they also form small pits, but the different proteins. And then there are some other pathways that are not really well known, and they are being sort of clubbed into clathrin and caveole independent endocytosis. So, all of this will result in some sort of a vesicle being formed containing particles that are membrane bound, and depending on what sort of particles you are using, what cells it is, what stage of this is being administered, these particles can escape through these vessels and cause intracellular delivery, especially in the cytoplasm, or these vesicles themselves can then be targeted to different organs. They can be targeted to mitochondria, they can be targeted to nucleus, they can be targeted to some other organ, or they might just be transcytosed. So, let us say if I want to cross a barrier with the cells on it, and if my particles do transcytosis, that means that this cell is going to take up this particle and essentially just throw it out on the other side. So, all of this is fairly feasible. So, now these particles are degradable and are carrying a drug that is extremely hydrophilic and would not have been able to diffuse through the cell membrane. Now, this drug can actually do that, right, because now this drug is in these particles which get taken up through these specialized uptake pathways, and now it is in the cell where it can get released. So, this is what we meant by intracellular delivery. And then since I mentioned here that these particles can actually escape from these vesicles, what is the mechanism through which they can escape from these vesicles? So, that is called a proton sponge effect, or at least one of the ways that we can sort of enhance this is using a proton sponge effect, and which is when particles are uptaken through endocytosis or phagocytosis. They typically get localized and are sort of entrapped in these endosomes and lysosomes, which is the machinery for the cell to degrade any kind of external particles or nutrients that it has taken up. And these environments are actually very harsh, they have very low pH, and lots and lots of degradative enzymes are there. So, if your drug is going to get released there, unless you want to target those locations, you want the drug to come out, because these are not conducive for the drug action, and it may even destroy the drug. So, what is done is to help the particles escape, effect which is called proton sponge effect is used. And so, what is proton sponge effect? So, these particles are designed such that they carry lots of secondary and tertiary amines, right. So, let us say if I have a polymer that carries lots of primary, secondary and tertiary amines, what is going to happen is now these amines have quite a lot of capacity to take up H plus ions, right, to take up protons. And as I just mentioned that these endosomes and phagosomes, so if this is a cell and here is my endosome, so the pH outside and the inside the cell is close to around 7, but the pH of this is now maintained at around 5 and it further decreases as it goes. So, these are early endosomes, and when these mature they turn into lysosomes, where the pH can drop down to 2 to 3. So, for the facilitation of this process to happen, essentially from here the pH is 7 goes to 5 goes to 2 to 3. The way the cell does this is it has lots of proton pumps. So, what it does, it takes up H plus ions or protons and pumps it in to the system, right. And obviously there has to be some sort of a osmotic balance, otherwise these vesicles would not be able to last, more and more water will also go in. So, now what is happening is, now I have put this polymer. So, now if I zoom into one of these endosomes, right. So, I have lots and lots of primary and tertiary amines that are present, they are taking up this H plus ion. So, they are taking up this H plus ions and essentially not letting the pH drop, right. So, the pH is still, let us say 6 at this place. Now, the cell does not like that, so it is pumping more H plus ions into it, and this continues till either you hit the saturation of your tertiary and primary amines, tertiary and secondary amines, and if we assume that there are so many of them that they will not hit that quickly, then what will happen is this H plus will continue to pump in. Because of this, now there is too much ions in your vesicles than there are outside. So, ions in here are greater than the ions outside, and so that causes an osmotic imbalance. So, now there is an osmotic imbalance, as a result of which the water from the surrounding starts to go in and sort of starts to maintain this osmotic balance. And as it goes in and in, there is some sort of capacity to which it can absorb the water, but eventually the pressure inside becomes so high that these vesicles just burst, right. So, once they burst in, the whatever particles are residing here, they come out and they are now in the cytoplasm, do not have to go through this harsh environment of 2 to 3 pH, and that is essentially is a proton sponge effect, right. So, I hope this is clear. So, essentially H plus is going in to maintain the charge, the chloride ion is also going in. Now, you have lots and lots of H plus and chloride ions that are present in your system, and because of that there is lot more ions in your vesicles containing these particles, and which then release which then causes the imbalance in the osmosis, and the water goes in to maintain that balance, and these vesicles swell and eventually they burst after a certain pressure is achieved. So, one of the polymer that is very widely used is polyethyleneamine. It is a highly positively charged polymer, just because there is lots of tertiary and primary amines, and again very widely used. And then there are other mechanisms. You can conjugate some peptides. This is mostly adopted from a viral strategy. So, some of the viruses, what they do is they have peptides that go and poke holes into the membrane. So, these peptides will go and sort of make a hole for which through which your things can escape and these vesicles may burst up. So, these are different mechanisms you can use to sort of utilize this proton sponge effect or these pore creating peptides to come out from your endosomal membrane. Okay, let us talk about some particle synthesis methods. So, first we are going to talk about chemical methods. These for polymeric particles involve some kind of a polymerization. So, you can take polymer in a confined state and start this chemical reaction. As the chemical reaction will proceed, more and more of them will cross link, and by the time the reaction finishes they end up forming a particle. For metal particles, reduction, oxidation, crystallization from salts is used quite a lot. So, you can get you can let us say have gold salt. So, you can have a Au salt, and then you can reduce it or oxidize it to basically return it to native state, where they will start to sort of interact with the other Au ions and form a particle, and depending on what concentration of the salt you have used, you can vary the size of these particles. And then there are lots and lots of physical methods much more widely used. These could be controlled precipitation of the polymer. So, basically you can have an emulsion process, and we will describe this in a little more detail as we go along, but you can have an emulsion process, and you let us say form these emulsion droplets with polymer plus solvent, but the solvent is volatile. So, it tends to just dry off, and once it does, this polymer concentration is going to start increasing, as well as this thing is going to shrink, and eventually all this polymer will just precipitate and kind of forms a physical cross link network, which will then release to particle, and whatever drug that might be dissolved in here just gets entrapped in here. And again, as I said, we will describe this process in more detail. So, solvent evaporation is again very similar method here, and there could be other methods that people are using. You can have a complex conservation. So, we discussed briefly about this during ionic hydrogels. The same mechanism can also happen at nanoscale depending on the concentrations and ratios of different things that you are using. You can actually actively remove the solvent rather than just relying on it evaporating out. There are processes for hot melt, spray drying, you can phase separate things out, and that may result in formation of nano and micro particles, or you can use salt to sort of induce this sort of separation or precipitation of the polymers. Okay, so let us talk about the solvent evaporation method, one of the very widely used method. So, first we are going to talk about single emulsion process, and so what is typically done is this contains two phases. You have aqueous phase and organic phase, and in aqueous phase you can have a distilled water with surfactant. In organic phase you can have some chlorinated solvent or maybe something else as well, also containing your polymer. Depending on whether your drug is hydrophilic or hydrophobic, you choose these methods. In this case it is mostly used for drug that is hydrophobic, and I will describe why. So, you add your drug into your organic phase, because if it is a hydrophobic drug it is only going to get solubilized into the organic phase, and then you emulsify it, and emulsify just means you mix them and give it some energy. So, when you give that, this organic phase does not want to interact with the aqueous phase at all. So, what it will do, it will first, if you do not give any energy, you will have them phase separate like this, where let us say if this is your aqueous phase or organic phase, or this could be vice versa depending on which one is heavier, and they will just separate out. But when I constantly give it energy and force it to mix, they will mix, but they will mix very reluctantly. So, what will happen is even though they have mixed, these two phases will not want to interact with each other. So, what will happen is depending on which amount is more, that will act as a bulk layer, and the rest of it just is going to sort of make these micro and nano phase separation, which is to basically prevent bulk of the organic solvent from interacting with the aqueous solvent, right. And so, the more energy I give, the smaller these droplets will become, and that is how they will sort of phase separate out. And now let us say this organic solvent is volatile or is evaporates at a very rapid pace. So, once this evaporates, what will happen is, first of all these droplets will shrink, and then eventually whatever the polymer is there will exceed the solubility limit, because the solvent is constantly evaporating out, and that will eventually cause this physical precipitation of these polymer molecules to result in nano or the microspheres. Okay. So, let us see. So, you one of the examples that is very widely used for this type of process is PLGA or PLA microparticles. They are again fairly hydrophobic. So, if we are using PLG, it has to go into the organic phase. One of the polymer that is one of the organic phase that is very used is either chloroform or DCM, dichloromethylene. So, this process is again referred to as oil in water, and the reason for the oil in water is because if the oil is in lower amount and this water, so essentially this oil droplets are in water. So, it is very famously known as oil in water or oil in water emulsion. This could also be oil in oil depending on what external phase you are using. So, you may decide to use, instead of aqueous phase, you may decide to use another oil, but this oil is immiscible with the other oil. So, in that case it will be oil by oil, but again this is typically not used for the applications related to the body, because we always want whatever particles that are made be able to interact with the water, and so for that one of the phase is typically aqueous. So, this, as I said, if I zoom into these small droplets that are evaporating solvent, what you are essentially having is these polymer chains essentially now when the size is decreased, these polymer chains are coming closer and closer, and then eventually they are just, there is no solvent, all you have is this polymer chains, and so these will essentially represent a solid matrix, right. There is no sort of capsule or hollow particles here, these are all solid matrix that are formed. So this process will result in a matrix type particle, not a hollow or a capsule. The drug must be soluble and dispersible in the organic solvent phase. So that is why I said it is used for hydrophobic drugs. If the drug is not soluble here, then it can't really go in. Only if the drug is soluble here, it will also be present in these droplets that are here. And then the polymer is typically dissolved in some volatile organic solvent. So as I said, one of the most commonly used is methylene chloride or DCM. Other solvents like chloroform and ethyl acetates are also used, but they have to be volatile, right. I mean, if they are not volatile, then this process will take forever for it to evaporate, and they have to be more volatile than the water, because you do not want the water to evaporate first. The polymer drug solution dispersion is emulsified again in large volume. So as I said, this is going to be in excess, whereas this is going to be limited. And then typically some sort of an surfactant is also added. So what the surfactant does, it just sort of localizes itself at the edge of these particles, because these surfactants, so they have domains that want to interact with the aqueous phase, they have domains which want to interact with the organic phase. So they sort of stabilize once these particles are formed. So you do not have to continuously give the energy for the during the evaporation phase. You can just give the energy once for a certain amount of time, and when these droplets have stabilized due to this presence of these surfactants like PVA, you can then leave it and do not have to continue to give energy to this. And then this can be stirred under reduced pressure and elevated temperature if you want to increase this evaporation rate, or you can do it at the room temperature and normal pressure as well to essentially let these particles harden and form these particles. Okay, so we will stop here and we will continue more in the next class. Thank you.